All right, so before starting this slide, I want to take a moment to thank Monica for giving me this opportunity to present to this GABA-A community. So my lab is at the Case Western Medical School. So we have been studying this GABA receptor since 2010, since we established our lab in Cleveland, Ohio. So we have seen the development of this field over the last 10 years tremendously, because when we started the lab at Case Western in 2010, that's only about maybe several GABA-A variants, but during the last 10 years now, probably you already know that there's over 1,000 variants have been identified in these GABA-A variants. So Monica asked me to give the community an update about the GABA-A variants. So this is a topic I'm going to talk about, the GABA-A receptor variants and the therapeutic strategies. Right? So brief background about me. So I'm Tingwei. So I'm now an associate professor in Physiology and Neuroscience department at Case Medical School. So it's good to be back to Los Angeles again. So because I got my PhD from Caltech about almost 20 years ago. So it's really good to be here to see really famous Freeway 110, because you know probably Freeway 110 is congested. Even in middle night, you need to maybe take 1 hour to go through that really heavy downtown Los Angeles. All right, now let's go with today's talk. This will outline my talk today. So first, I want to give you a physiology about the GABA receptor. Then we try to, because when we study the disease, we want to study this physiology, what is known, what is for the phenotypes. Then we want to study the disease-causing mechanism. After we get the details about the disease-causing mechanism, we can design different therapeutic strategy to target different variants. So I will follow through this thread for my talk. So I think Dr. Mary Chebby gave a really nice introduction about this ER balance. I don't need to take too much time here, right? So because the brain is a tag wall between inhibition and excitation, the loss of inhibition of the GAN wall excitation is traditionally known to cause this epilepsy, right? So also you probably will recognize some of this middle figure is already presented by Dr. Paul Davis earlier today. So you can see that the GABA field has been really unified here, right? So we know that about the GABA receptor is really important in regulating this. Let me see. Regulating this excitability for this pyramidal neuron, right? So in pyramidal neuron, we will have input from both the GABAergic interneurons as well as the excitatory input from the glutamate site, right? So GABA receptor, probably you are already pretty familiar, is pentameral receptor, right? So in the synaptic side, it has the alpha 1, alpha 2, and alpha 3. Surely Paul Davis has some different slides showing that alpha 4 will have different actual synaptic site. And also people have known for a long time GABA receptor has 2 binding sites for GABA and also 1 binding site for the benzodiazepine, which is as actual cellular interface between 2 subunits. And also during the last several years, due to the development of the cryo-EM field, people have get really high-resolution structures for heteropentameral receptor, including alpha-1 beta-2 gamma-2 and alpha-1 beta-3 gamma-2 subunits. And the cryo-EM structures have confirmed all the previous predictions about the details about GABA binding site between beta-alpha, and the benzodiazepines binding site in the alpha-gamma-2 interface. Okay, so this is a background. Then I also want to talk to you about that. You know that from here, GABA receptors that really need to be present as a synaptic plasma membrane. That's their functional site, right? That's the location GABA receptors supposed to do if they are functional, right? But inside the cells, there's a biogenesis pathway for these GABA receptors, right? So probably you know proteins are synthesized in the nucleus. Upon synthesis, protein need to be enter this organelle called the endoplasmic reticulum, or abbreviated as ER. The ER is a protein factory that are responsible for the folding and assemble all the membrane proteins, including GABA receptors. Only after the folding and assemble of these pentameral receptors, the GABA receptor will be able to traffic from the ER through the Golgi and reaches the membrane. It is only after that reaches the membrane that will be functional, right? So if something goes wrong with the protein processing process inside the cell, GABA receptor will not reach the plasma membrane. Therefore, they will lead to their functional deficiency. This is also one of the underlying hypothesis, working model for how we can design the therapeutic strategy for different GABA receptor variants. Okay. All right. So this is also really important slide. This recapitulate what I just mentioned, but it's also important. It's a really cartoon. This will draw your attention to this protein folding and trafficking pathway. As I mentioned, GABA receptor need to assemble properly into this pentamer before they will be able to traffic out of the ER and reach the membrane. The GABA receptor individual subunit then to fold and assemble well inside the ER, the organelle in the cell, right? If something goes wrong with the protein folding or protein assemble step, the proteins will be retained inside the cell. Therefore, they will not be able to traffic out of the ER to the plasma membrane. Also, probably you know that many of the GABA receptor variants are resistant to current drug treatment. I think one really important disease-causing mechanism is that those variants will cause retention of the GABA variants inside the cell. Therefore, they will not be able to be present on the membrane. That's why many of the miracle drugs on the market which can only function when the receptor reaches membrane, they have no influence on these GABA variants. And also now, probably you can appreciate that there's over 1,000 variants for the magic subunits, GABA receptor, alpha, beta 2, beta 3, and gamma 2 subunits. So now I think that many groups, including Nathan, Mary, and Dr. Moll, and the Katie Khan's group, including our group, have been doing a really nice characterization about the different variants among this GABA receptor spectrum. And also in my group, I have some really talented grad students and postdocs and senior scientists. They are working on different variants for these different GABA subunits. Bianca, who is happen to be in the origins, she is working on the alpha-1. Dr. Megan Wang, she is working on alpha-1. Chelsea is working on beta subunits. Mani is working on the gamma-2 and alpha-1. You can see that my group has been working on alpha-1 subunits really extensive during the last 10 years. So we have some really interesting, really nice data accumulating about the GABA receptor variants, how they will cause their functional deficiency. This is what I can show you about some of the prediction from the NIH Tingwei database, right? So this is a sequence alignment about different subunits for GABA receptors. The red means that in the Tingwei annotation it's pathogenic. The yellow means that's uncertain, right? So immediately you can find that for the transmembrane helices, it enriched a lot of the pathogenic variants. It's not very difficult to understand because the GABA receptor has a transmembrane protein. The transmembrane domain need to be inserted into the lipid bilayer really efficiently. If something goes wrong with that transmembrane domain, it has a high chance to lead to functional deficiency. Surely, probably in the GABA field, people know that TM2 faces the central power region, right? So any mutations in the TM2 region, they will tend to generate the gating deficiency, lead to really severe functional deficiency. Surely, I think you can also appreciate that many of the residues in the ER lumen, especially in the loop areas, they also have the tendency to lead to functional deficiency. The work from our group and also from Katie Khan's group, we also demonstrated that many of these variants in this ER region will lead to their protein traffic deficiency, means that these variants will be retained inside the cell, leading to their extensive degradation. Therefore, there will not be sufficient amount of proteins on the membrane for their function. All right. So, and also, I want to compare this GABA variants to other well-defined protein folding field. This means the CFTR field. In the CFTR field, people know that the misfolding of this CFTR will lead to this cystic fibrosis. People can design drugs that can restore the trafficking of this misfolded CFTR to the cell surface as a way to effectively treat this cystic fibrosis, known lung disease, and also Vertex pharmaceuticals has been very successful in designing those kind of drugs to treat cystic fibrosis, which has been developing into a multi-billion-dollar company. So for us, we try to borrow the successful story from a CFTR field, try to see how we can contribute to this GABA receptor field. So this is for these different GABA receptor variants, we know that we can categorize these variants according to their molecular functional deficiency. The first one is called the proteostasis deficiency. This means that if this variant occurs, the protein will not be able to travel to the membrane for their function, right? So the second one is that about the electrophysiology deficiency. This means that this variants will cause their gating deficiency, leading to their like either kinetic deficiency or some of the agonist potency deficiency. The third one is called the nonsense of friction mutation. These variants will cause their mature truncation of the protein, therefore they don't have full-length protein for their function. All the rest goes to the class 4 variants. Okay. Then this is the system we are using. Basically, we do cell physiology. We use both established mammalian human HEC293 cells for our initial proof of principle study, and now we are also developing human iPS-derived called neurons as our native states to study GABA receptor function. I know there's many patients in this group. It's really important for you to contribute some of your samples, so people then combined brain will be able to convert those blood samples into iPSC cells. Then research lab will be able to get these iPS cells and differentiate into cortical neurons, right? So we have been studying this using this iPS cell to study GABA receptor for the last several years, and we have achieved really nice progress on this front. Bianca is developing this iPSC pretty nicely. Basically, she can differentiate this iPSA into cortical neurons. After differentiating into cortical neurons, we will be able to use electrophysiology, proteomics, and confocal immunofluorescence to monitor trafficking function and the disease-causing mechanism for this disease-causing variants. So this is what Bianca bring a poster with her. He will present this Sunday. So you are welcome to join Bianca to ask her any questions. Basically, he showed that we can. This is the Y-type iPSC cells. This is disease variant carrying iPSC cells. She can induce this iPSA into this umbilical body. This is a stage that is important to convert this iPSA into neuroprogenitor cells. After generating these neuroprogenitor cells, it can be further mature for neurons. Bianca used different markers to label different stages along this process. But this iPSA work is pretty expensive and really time costly, right? So because you can see the induction into NPC cells will take 10 days. Then the mature into neurons will take another 3 to 4 weeks minimum. So this is a really lengthy process, and also finally we need to generate a healthy neuron for the following characterization using the method I show here. Basically, for the electrophysiology, there's 3 methods we are using now. On the top left, you probably, people can recognize this traditional patch clamping setup, right? So it's a really huge system. People just use this really time consuming and also technical challenging. But people can use this one to patch HEC cells, neurons, to study their electrophysiology properties. This can be done. And also in our department, we purchased this so-called automatic patch clamping system. This system is amazing because this will be able to let us to record the occurrence from HEC cells expressing GABA receptor within really short time period. For example, we use this 96-well based plates. We put the cell into this plate, then the cell will flow through across this route. Then for each trap, it will be able to get 20 cells. We will be able to patch those 20 cells in each wells. And also, all this process happens really fast within 10 minutes. We can record this really nice E-safety curve within 10 minutes after we put the plates inside the automatic patch clamping. This is really powerful stuff because this enable us to record many of the variants in really short time. Usually, when we try to get E-safety using HEC cells patch, probably you know it will probably take weeks. But with this automatic patching system, we can do it within 1 day. So this is really powerful stuff. And also, if we want to study neurons using iPS-derived cells, we need to monitor the overall neuronal excitability, which is important in the GABA variance field because we want to monitor the final excitability of neurons. In addition to this individual function of this GABA receptor. In this case, we use this so-called MEA plate. It's a really nice instrument. We will be able to get this neuronal excitability to compare the variance with the wild type as well as the drug effects, how the drug will influence overall excitability. In addition to this patch clamping setup, we have been routinely doing. Okay, we have all this routinely operating in the lab. Okay. I know I'm out of time. I just go through this very quickly because this is just the GABA receptor variance characterization. For the alpha-1 subunits, that's over 300 now, right? So we cannot characterize all the 300. In this case, we selected 8 representative from this GABA variance. How we choose this representative? One is that we select the variants according to their pathogenic probability. We can predict this probability using these AI-based tools. It's really accurate. And also we select the variants in different space distribution along the 3-dimensional structures. We choose some variants in the ER-Lumen domain, some variants in the transmembrane domain. This is the 8 variants we characterized recently, and also using the automatic patch clamping, we were able to get all these EC50 curves for these variants as well as the peak current amplitude for these different variants. And also, we focus on alpha-1 subunits. Somehow for this select 8 variants, alpha variants, all these variants need to this loss of function according to the peak current, right? So surely we can find that many of the transmembrane variants are more severe than the ER-luminal variants, right? So this is just the way it is. For GABA receptor, we also try to monitor their surface trafficking. We know that the majority of these 8 variants we selected have insufficient membrane stuff, and also this is probably last several slides. This is we also try to how we can create the folding of these variants into their to restore their function. So basically, we use 2 strategies. One is called protease regulators. So these are general protein folding factors. We have collaborating with Katie Kang as well as my postdoc mentor Jeff Kelly at Scripps. We use different like unfolded protein response. We also did screening, identify different drugs for this purpose, and also we use the so-called pharmacological chaperone that direct GABA receptor binders, right? So this binder direct GABA receptors binders, they are specific, so they will be able to stabilize these variants for their functional rescue. You just need to believe me that we did a screening and found this really small, really nice histopathology molecule for their functional rescue for all the variants we selected so far, right? And also this is just demonstrate that we use iPS-derived neurons to monitor the surface trafficking, to determine how this histopathology will be able to restore their surface trafficking, right? So I think that's it tells us that indeed now we can see that reduced trafficking is loss of functional GABA receptor is a major disease causing mechanism for GABA receptor variants, at least for the alpha-1 we have been demonstrated. We are also working on the beta and the gamma-2 subunit to see whether this is still the case. On the second one, I hope that I show that restoring protease stasis pharmacological corrects the surface trafficking and thus function of these GABA variants. Finally, several lead compounds such as histopathology and dihydroergocristine, they were really promising lead targets that can be further developed to treat these GABA variants. Okay, finally, thank you very much. This is really important because this is the people who do the work, right? Because Bianca is the person here, she is working on the alpha-1 subunits. Megan did all this pharmacology chaperone work, and also this is our group inside the lab. This is our group of, when we did the group retreat in our farm, and also we have collaboration with Jeff, Luke at Scripps, with Katie Kang and Vanderbilt, some other really nice collaborators at Case Western. And finally, I want to thank the funding from the NIH. So because without funding, we cannot do anything. Okay, finally, thank you all for your attention.